Promising development in the war on cancer. Researchers are now testing some designer drugs which disrupt the growth of cancerous cells without harming healthy ones. Here's NBC's Dr. Ian Smith.
2 more volunteers, please.
Each morning during her junior year English class, Virginia Garner quickly downs the pills she says have made life worth living again.
The joy of my life right now is that I can do all the things I used to be able to do, and also have some hope that maybe I'll be around for a lot longer.
3 years ago, Garner was diagnosed with a relatively rare and often fatal type of cancer called chronic myelogenous leukemia, or CML. A single genetic defect causes her white blood cells to multiply out of control. But by participating in a clinical trial, she gets these experimental pills, a drug called STI 571 that takes dead aim at that genetic abnormality. Dr. Brian Drucker led the clinical investigation of this new drug and has reported remarkable success so far.
We had a perfect target. We knew what caused the disease. We knew what caused the white blood cells to grow uncontrollably, and we knew exactly what to go after in designing something to shut down this leukemia.
Dr. Richard Klausner, director of the National Cancer Institute, says this kind of precise targeting holds the promise to revolutionize cancer therapy.
The move from empiricism, which is sort of guessing about things and hoping you stumble over something, to really designing, going after the targets you know are there, is a profound paradigm shift.
STI 571 is just one of several experimental smart bomb drugs that take this new targeted approach. Unlike radiation and chemotherapy, poisons which kill both cancerous and healthy cells, these new drugs are designed specifically to interfere only with those biological processes that turn normal cells into cancer. They're possible now because after years of laboratory research, scientists have a much better understanding of how cancer behaves.
Instead of just thinking about a cell that's growing, finally we're seeing the machinery, the details. It's like opening the hood of a car, and you see it doesn't just move, there's an engine, and there's an electrical system, and we see how the parts work.
In cancer, those inner workings involve a series of cell signals programmed by genes. All cells multiply when a protein called a growth factor binds to a receptor on the cell's surface, triggering a release of chemical signals inside the cell. These internal signals cause a chain reaction that ends in cell division. But in cancer, this process can become overactive, leading to excessive multiplication. Some of the new drugs work by blocking the surface receptor, preventing the entire process from starting. Others, like STI 571, interrupt the chemical reactions inside the cell before the cell can divide.
Grab a seat.
For Sam Johnson, whose advanced colon cancer spread to his liver, a different type of experimental drug takes on a different molecular target. SU5416 binds not to cancerous cells, but instead to the growth receptors on nearby blood vessels, preventing new ones from growing toward the tumor.
Tumors growing very quickly, very rapidly outgrow their blood supply. If they cannot create new blood vessels for themselves, they'll starve and they'll die.
Doctors are very encouraged by the early results of this study. Sam Johnson, after almost a year on the new drug combined with traditional chemotherapy, has had his tumors stabilized.
As Dr. Rosen told me, staying the same is great. If my tumors don't grow, they won't kill me.
A generation of cancer research leading to a new generation of cancer drugs, still in the testing stage, but providing hope for millions of people by homing in on the biochemical malfunctions that turn normal cells into cancer. Dr. Ian Smith, NBC News.